All right, good morning and happy April 21st. We're going to get started with the last part of Unit 4, and I'm actually going to do this in two videos for you so that you can break them up so any one video isn't too long. So we're going to start with, what happens if there's an error in meiosis, and we've talked a little bit about that before. And then we're going to go through introducing evolution, talk a little bit about one of the essay questions, and that's where the first video for this part will stop, and then we'll pick back up. All right. So in class previously, before we moved completely online, we talked about, well, what are some things that can happen if cell division doesn't go like it should? And one of those things that we came up with is either too many or too few chromosomes. And so we're coming back to this idea and giving it a specific name. So if there's an error with cell division, more specifically with meiosis, it's called non-disjunction. And that just means that there's an error in meiosis through no one's fault at all. It's just that the process has some risk of error. And so one of the common things that can happen, especially if the parents are 35 years or older, is that there's an extra chromosome 21. And you can clearly see that down here on the karyotype. So the genetic testing could come back that there's an extra chromosome. And with trisomy 21, we call this Down syndrome, and there's a wide range of severity for this particular medical condition. With medical advances, we know that these individuals can definitely have very full lives and are living to be older than they once did. Average lifespan still is about middle age. So that non-disjunction usually ends up with some gametes, so some egg cells or sperm cells that have too many chromosomes, and other cells that have not enough. And so once fertilization happens, those zygotes aren't going to have the number of chromosomes that we would anticipate. So we're talking an extra one, 47 for humans, or 45 for humans, with 46 total being the expected number. So to get a visual of how this impacts your normal meiosis occurring up here at the top, we have 23 chromosomes in the egg cell, 23 in the sperm cell. Putting those two together, and we get 46. Again, if we're talking about humans, other organisms will just have a different number there. The pattern is the same. If non-disjunction occurs, we may have a cell that has too many or not enough, and then that zygote goes on to have too few or not enough. So I think this is a great diagram to take a look at. Number one, to double check your understanding of meiosis, and number two, to add that next layer of detail that non-disjunction ultimately has an impact on the fertilized egg. So non-disjunction can also happen with the sex chromosomes, and I've been alluding to in a few of the videos that we'll talk about variations of what we would consider to be XY or XX. Socially, that's what we talk about, and I think that's what we think is the only option out there, and that's just not true. There's a lot more gray area to the sex of organisms than we actually talk about in society. And so this non-disjunction can occur with the sex chromosomes, meaning having too many or having not enough. And so what can happen? We can end up with XXY, and so that's Klinefelter syndrome. 1 in 2,000 births are actually the XXY, and just think about, what that means. For every 2,000 people, one person would be XXY. And And they might not necessarily know that they have XXY, but if this individual would go on to want to have children, you can see one of the effects is that they would be sterile. So they could go through their entire lifespan and not necessarily know that they have XXY. And generally speaking, they would have features that we would consider more feminine, but still present as a male. We could have XYY. Again, 1 in 2,000 presents as a male, and this person probably would never know that they had the extra Y chromosome. Growth and body size really is the only effect that we see, so no real reason why someone would know that they had the extra Y chromosome. We also have triple X, so 3 X's, and that female would present as normal. So again, another variation that that person would really have no reason to know that they had a sex chromosome abnormality. And then with Turner syndrome, this is the only case where having 1 less chromosome than anticipated is not fatal. So the only case that 45 chromosomes can lead to relatively full development. This person probably would know that something is going on 1 in 5,000. So out of all of these, this is the least common, with triple X being the most common. But this individual would be a lot shorter, probably have some extra skin on the neck, would not be able to have children, and then the sex organ development would not proceed as anticipated. Excuse me. So these are just a few of the possibilities of non-disjunction with sex chromosomes. And because we know sex chromosomes impact both estrogen and testosterone production, just kind of think about how these variations can lead to differences in hormone levels, and kind of what else that could lead to in addition to the effects that are listed. All right, so headed into talking a little bit about evolution, and we looked at this after meiosis, because meiosis is what gives us genetic variation. And our definition of evolution is genetic change over time. Some textbooks do a really good job of laying out that definition, and other textbooks could be a little bit more explicit. But I put that in a different color because I want to make sure to highlight that we have a common understanding that when we're talking about evolution, we're looking at genetic change over time, and ultimately those genetic changes can be connected back to meiosis, and the variation that we see with meiosis, with things like crossing over, random fertilization, mutations that occur. So ultimately, that genetic change is going to be how evolution occurs. And sometimes that can just get a little bit muddy, but I want to make sure that we're talking about the same thing. So when we're looking at populations, so while we may use the word evolution in a general sense to say, oh, I evolved over time, what you really mean is I changed over time, ideas changed over time. That's not how we use that term in science. So with evolution, we're talking about populations, so groups of organisms changing over time, and that time usually is marked by generations. And so ways for that change to occur, we talked about DNA mutations. That gives us different sequences of A's, T's, C's, and G's. Selective mating, so meaning that mating is not random. Migration between populations, so individuals moving into and out of populations. And then this idea of natural selection where there are environmental pressures. Excuse me. Environmental pressures where certain organisms are going to have an advantage or a disadvantage to survival, and the ones that have the advantage for survival will reproduce and then pass on their traits that are hypothetically more adapted to that environment. So individuals can't change those characteristics, but they can be passed on from generation to generation. So when we think about descent with modification, what this means at the intersection of any line here is that there was a common ancestor that was not a mammoth and that was not an elephant, for example, but a different organism that they both relate to. And what happens is there's some sort of speciation event, some sort of external pressure that causes the two populations to not be able to reproduce with each other anymore. And that's really the measure for how organisms come about to not being able to reproduce, and we call that speciation. At the end of this unit, we'll come back to that idea and talk about, well, what are some of those speciation events? What would cause that to happen? One, I think, picture that we could get in our head is that maybe there's an earthquake and there's a big hole in the ground now, and that crack is so big that two populations can't interact with one another. So physical separation is one of those pictures for a speciation event. So a required essay question for this exam. The first essay question will actually be a genetics problem that you'll need to solve, just like a lot of the activities. And the second one is you're talking with a friend who asks about what you're learning in biology at Central Carolina. You tell him or her that you recently learned about evolution. Your friend responds, I don't believe in evolution. Describe how you would convince your friend that evolution is a real scientific phenomenon and include your understanding of the term theory and evolution, and use at least 3 examples or pieces of evidence. So what I'm looking for full credit here is for you to describe the term theory, describe the term evolution, and then we're about to go through the different examples or pieces of evidence that you can use, and that will get you full credit. All right, so the first piece of evidence that you could talk about is the fossil record. So fossils require bones, and we know that not all organisms have bones. Things like insects don't have that hard shell to leave any trace behind. So while fossils are a great start for the different type organisms that were alive at certain time frames, it's by no means exclusive. And so if an animal or an insect doesn't have a hard body, doesn't have a skeleton, doesn't have a shell, then they're not going to leave evidence that they were actually there. So fossils are a great place where we can get information, and based on the location that the fossil was found and also the different layers of sediment, they can make connections as to when that particular organism was alive. They're able to also do that through radiometric dating. So radiometric dating is your second piece of evidence that I talk about. And this just means that we can take fossils and then give them a date. And so we know that at certain periods of time, there's a ratio of carbon-14 to carbon-12. And we also know the rate of decay of carbon-14 over time. And so we can actually use that ratio and use the decay to predict the time, or the age of a certain layer. And so we can see this as a graph. So percentage as a living organism. And so that's our basis. So knowing what the ratio is when an organism is living, and then looking at what our sample is in comparison. So our timeline down here, you can see the unit is thousands of years. So we're talking about a pretty big time frame here. Another form of evidence that you could talk about is transitional forms. And this just means that there are organisms that have features that they necessarily use, but they can be a connection between one organism and another. So for example, a lot of these whales had hind limbs, and you can see all of the bones down in there. So they didn't use them as limbs, but we know that organisms that aren't quite so old actually ended up on land, and those limbs were useful at one point in time. Another thing you could talk about is biogeography. So 10 million years ago, there's evidence of one big supercontinent. And so everything was connected at one point in time. 50 million years ago, we can see Australia breaking off over here, and then what we know as present-day Australia. And so the kangaroo, the koala, these are organisms that are only found on the continent of Australia. So meaning that the distribution of species provides us some evidence for how things have evolved over time. There is one marsupial that we find in North America. If you want to pause and try to think about what that marsupial is. Sometimes this organism can be considered a nuisance, but the possum actually has a very important role with making sure certain pests are controlled. So marsupial meaning that the organisms carry their young in a pouch, and the possum is the only organism in North America that does that. Another thing you could talk about is comparative anatomy. So this gets back to comparing the bone structure of organisms. So even though the forelimbs here have very different purposes, the thin wings of a bat compared to the porpoise compared to the human, we can compare and color-coded that, okay, each of these three animals has a hand, it has wrists, it has two bones in the arm, it has a humerus or upper arm, and then it also has a scapula or shoulder blade. And so that similarity gives us evidence of connection. We can also look at comparative embryology, meaning that if we look at the embryo of a chicken and compare it to a human, there's really not a ton of difference. Humans do have tails as embryos, and we also have pharyngeal pouches that eventually develop into lungs. So the fact that at a most basic level after fertilization, embryos look very, very similar and generally are of similar size. And you can see this in a little bit more detail. So we've got a fish embryo, a reptile, a bird, and then a human, so a mammal. And we can see that they all look very, very similar. You would have to pick out just super small nuanced details to tell them apart. And then the last piece of evidence that you can talk about, for me, it's the strongest piece of evidence, but the fact that we can compare DNA and do a base by base match provides evidence of relatedness. So when we're doing comparisons, humans have over 98% of the exact same bases compared to chimpanzees. So that means base for base, A's, T's, C's, and G's. So that doesn't mean that we evolved from the modern day chimpanzee. It means that there was a common ancestor, and that our DNA matching is evidence of that. All right, so back to the required essay question to summarize, making sure that when this question comes up on the exam, that you explain what a theory is. So at the very beginning of class, we talked about the difference between a theory and a hypothesis, and the theory of evolution having multiple lines of evidence and thousands upon data sets to support it. Your understanding of the word evolution, and then you can pick 3 of these different pieces of evidence to explain. Make sure you fully explain it and describe what it is, and if there's something additional specific that you can add to it, that would be great. So you can choose between fossils, radiometric dating, transitional forms, biogeography, comparative anatomy, comparative embryology, and then talking about the DNA similarity. All right. Thanks so much. Have a great day.